The structure of the small intestine is key to its function. If we looked at a cross-section through the small intestine, in most regions there are abundant folds. Each of the folds has many folds. If we looked at just one of those areas, we'd see down here at the bottom a connective tissue layer, the outermost layer of the intestine. Then a longitudinal muscle layer running lengthwise along the intestine, and circular muscles that run around the intestines, around in both directions. When these contract, become shorter, they make the whole intestine narrower. The longitudinal muscles and the circular muscles together can cause churning of the food or segmentation, breaking up the food particles. I'll show one of these villi, and down below is a gland common at the very beginning of the intestine. That gland is lined by cells, an epithelial layer of cells, secreting two things: sodium bicarbonate as a buffer, and cells that are secreting mucins in a vertebrate. That sodium and bicarbonate and the mucins, which form either long interconnected tangled chains or meshes depending on the different types of mucus. The sodium bicarbonate is neutralizing protons, H+, from the stomach, and forms the weak acid carbonic acid, H2CO3, and not as the free H+ and bicarbonate. And the mucus helps to lubricate the food and also creates a layer that protects the cells in the villi. The villus cells themselves are also secreting mucus. There are goblet cells often that secrete both tethered mucus and free mucus in multiple forms. Now let me show you a histological section of just a portion of the lining of the intestine. We'd see something that looks like this. So here's one with a large number of what are called goblet cells, because these look vaguely like a goblet. They're secreting mucus. The mucus is in two forms, tethered and free. There are also absorptive cells with what's called a brush border, because under high magnification it looks like a brush. Here is a scanning electron micrograph, just one individual cell. These things you can't see very well are mitochondria. These long, narrow projections are microvilli. Movements will allow material to move so that this entire surface area can absorb material. Tethered along the border in the mucins are many terminal enzymes of digestion that are doing the final conversion of small fragments of proteins, just a pair or a few amino acids long, moved via secondary active transporters, would be moving amino acids and glucose with sodium, powered by the sodium concentration gradient. In the interior of each one of these villi would be an arteriole going up with capillaries branching off and a venule coming back, bringing blood up, and the vein with products of digestion, things that have been moved into the cells and then into the capillaries and into the veins. Many substances don't end up in the capillaries or the veins. There are also lymph vessels. Fats in particular are processed into chylomicrons, into structures that end up moving into the lymph, and they're transported through lymph vessels. The output from these venules eventually is sent via the hepatic portal vein to the liver for processing. And the liver does preliminary processing of digestive contents and also screens and checks for invasive bacteria or viruses, and the liver contains enzymes that will break down many toxins. Here's an overview.